Now, tonight's really all about you, because I really love people. And I just want to ask you, when you woke up this morning, how did you feel? Like, were you thinking, I have the best possible life? Can't wait to get today started? You know, like, wow, I live in Australia, particularly Adelaide. I mean, Adelaide's gorgeous. I was running along the river there this morning thinking, wow, this place is really nice. Or were you thinking more along the lines of, oh, God, I've got to get the kids lunch and my boss is really annoying me and I have all these deadlines and, oh, my God, my superannuation isn't as high as I thought it was going to be and, God, the house prices, what's going to happen with them? If you're about 90% of my audience, basically people respond more likely to that than to the opposite. So my question to you is, do you know what's happening inside your brain when it's stressed out, or even your body for that matter? So isn't that remarkable that after 25 years or more and hundreds, thousands, millions of papers, that we don't really know what's happening inside our brain, or we can't really see it, and that's part of the issue of why I came out of my research lab. So what I'd like you to do now is just to put your arms up, stretch them high. Oh, doesn't that feel good? Oh, take a deep breath. Oh, well, you know, I just biohacked your brain. In fact, I biohacked the emotional part of your brain, which is the oldest part of your brain. So I wasn't always going to be a neuroscientist. No, I'm a country girl from Nanango. And I was actually writing an honours thesis on why people buy cough and cold medications in a pharmacy using logistic regression. I know, hold on to your seats. It's thrilling stuff. And about halfway through that thesis, basically I get a phone call from my mother and she says to me, Selena, you've got to come quickly, something happened with your sister. So for the next 3 weeks, my mother and I spent 3 weeks visiting my sister in a lockup ward in Wollston Park, and her treatment was a straitjacket and an overdose of haloperidol. And there was one particular moment where I was sitting in this room with the doctor and my sister. She was basically stiff as a board and catatonic from the medication, because basically she was a little girl with not very much happening, except she told someone that she was hearing voices, and I highly recommend you don't do that. No, just joking. Anyway, I shouldn't joke about it, I know. But anyway, so during that moment, it became very clear to me why people buy cough and cold medications in a pharmacy didn't really matter anymore, and how the brain works. We don't seem to understand how that works very well. So basically, I want to dedicate my lecture tonight and my plenary lecture tomorrow to the ANS to my sister, Francesca Hokart. She's the reason I'm a neuroscientist today. So I went back and I did my PhD. I didn't know how to use pipette. I didn't know how to do surgeries, brain surgeries. And for the next 10 years, I did a PhD in Queensland, and then I got trained in an amazing neuroscience institute at the John Curtin School. And many of those amazing scientists are being awarded at this ceremony this week, I noticed. But I had a baby in '97, and then I was pregnant with my second child. And I was sitting in my office and I became very enamored with this work that was being done in San Francisco. What you're seeing on this picture here is a neuron, and all of the reason it's lit up is because they're receptors, and at that time in 1999, they could visualize those receptors using this thing called green fluorescent protein. And at that time, where I was working, I didn't have the technology or capability of doing that. And so like you do, you move to San Francisco with 2 babies, you know, and you start a lab there, of course. You know, that's what you do. Anyway, and I was very fortunate to get a position at this place called the Ernest Gallo Clinic and Research Center for Alcohol Addiction. And Ernest Gallo started this addiction center. He actually planted the first grapes in the US. And then, much to his lawyer's chagrin, he decided to start an alcohol addiction research center. And so every year he was the chairman of our board, and we had to say what we did lately. But one thing that he did that was remarkable in the 80s, when he first came up with this idea, most people were studying the impact of alcohol on the liver. And he kept saying, it's not my liver making me drink. So he actually turned the centre into a neuroscience centre where we had to understand the neuroscience underpinning alcohol addiction. And then also we were funded by the state of California, so we had to actually come up with a treatment for alcohol addiction. And the director at the time said, oh, we'll do it in 5 years. Right, okay, so they actually approached me about a year after I started there to become the director of medications development. They gave me $1 million and 1 year to come up with a treatment for alcohol addiction. And because I'm a country girl from Nanango, I don't know anything, so I say yes. And so then I literally did not know anything. I couldn't even spell neuroscientist when I started that. Anyway, so the first thing that I started to work on, I put my pharmacist hat on, and I want to tell you, during this, just before this time, I was totally focused on mutating amino acids in the C-terminus of the mu-opioid receptor. Are you now excited? So that receptor is where morphine binds to. And the person I was working with was the person that was studying those receptors going inside the neurons. And I just loved it. I was like in fairyland, because over there you get your antibody overnight and all of these things. So I had 1 million experiments set up on my lab bench. But I was sitting there going, only 5 people are reading my papers. How's this going to help my sister? So anyway, I got approached to do this particular job, and I took it on like everything else, like seriously. And so what I noticed was, because of my pharmacist training, I noticed that there was 25 years of research that had shown the link between people that are dependent on alcohol. They also smoke, but people that smoke aren't necessarily dependent on alcohol. Now, what's the common neurobiological mechanism driving that is the fact that nicotine binds to the neuronal nicotinic receptor. So when you smoke, the nicotine binds to that receptor, releases dopamine, and makes you feel good. Right? So that's what alcohol is doing in synergy with those two things. Now, I got really lucky. So the day I accepted that position, the next day I flew to Boston to give a talk at the Gordon Research Conference on my previous work. Who was in my session but the person, the scientist that actually discovered varenicline, which is a drug called Champix in America and Chantix in Chantix in America and Champix in Australia. And the data he presented during that session just threw me out of the, I just couldn't believe it, because the drug wasn't metabolized by the liver. It had been used by 6 million people. And I'm thinking, oh my God, I've got to get that drug and get it into my lab to see if it can work for alcohol addiction. Anyway, but he said to me, Pfizer will never give me that drug because I'm not allowed to contaminate their findings with my dirty alcohol studies. And so what I did was I phoned them for 2 weeks, like for 2 years. And so therefore my lab was the first to actually get this drug into UCSF. So what that meant was, why we were the first to publish is because my lab seriously started working on the animal models of alcohol addiction. Because when I started, we were actually studying social drinkers. We weren't studying people that had been drinking a lot of alcohol. So I actually changed the animal models, which meant that when we got the drug, it actually worked. And this happened very quickly. And so because it had been approved by the FDA, we were able to fast track that into clinical trials. So in this picture here, you can see that this is a very small proof of concept clinical trial demonstrating how the drug can reduce alcohol-induced activation of the part of the brain that drives the reward center. The other advantage, obviously outside being on Good Morning America, because we found one drug for 2 addictions, is that we could do very small-scale human clinical trials. Now, we got some good results. We've got about a 10% response rate. I know, hold on to your seat. It's really big. And at the same time, I was just, the lab next to me was a young woman who was working with the godfather of neuroplasticity. So neuroplasticity is the ability of the brain to change with training and effort. She worked on songbird, singing in birds and watching how different parts of the brain will change. And she joined the Gallo Center and set up her lab next to mine. And one day she's passing me in the corridor and she says to me, Selena, do you really think that medications are the be-all and end-all? And I said, yes. And so she took me up to her lab and she said, please come up and have a look. And she actually had adult animals on treadmills, and she had the capacity to actually map the changes in the synapses happening in the top part of the brain in adult brains that were exercising. So she planted a seed in my head on my transition back to Australia. This is research coming out of my current lab in Brisbane. So what I want to point out here is that alcohol, sugar, and junky food and stress is actually changing the physical and chemical structure of the brain. I think that's really important to just think about for a second. When I think about that, it blows my mind. So that means you coming here tonight and thinking about and listening to what I have to say, if you pick up on just one item and take it and apply it to your life, your brain has the capacity to change. And when you understand that, it really is, in my view, revolutionary. So this is just demonstrating. This is the prefrontal cortex. This is the front part of your brain. It's a part of your brain where impulse control sits, where decision making in the short term leading to long-term goals. So for instance, how to resist the second donut sits in this part of your brain. So that's what addiction is. It's an impulse control disorder. The reason why stress and addiction are linked is because stress takes down the prefrontal cortex, which makes impulse control worse, which I'll talk to you in a minute. So on my move back from California to Australia, we had set up an experiment where we were trying to understand how does alcohol change the brain at the molecular level, at the biochemical level, and we were actually specifically studying neuronal nicotinic receptors. I was working with the expert at the Stanford Research International. We set up this experiment where the animals drink alcohol and our control was sugar. And we had another one, fortunately. But anyway, she rings me. I'm sitting at my desk in Brisbane. She goes, "Selena, you won't believe this. Sugar is changing those receptors in exactly the same way that alcohol and nicotine are." And I'm like, oh my God, that can't be true. There's no way that can be true, because that's the control, right? So my poor PhD student in Brisbane, because I didn't believe it, he had to go and replicate it all, and then we had to do all the experiments over a number of years. And so we end up showing that just like varenicline, which binds to nicotinic receptors, this particular drug, Tabex and Champix actually, can reduce sugar consumption, right? Doesn't that blow your mind? So what that means is prolonged overconsumption of sugar actually changes the physical chemical structure of your brain. It activates the same addictive pathways in the brain that alcohol and nicotine do. So that means sugar, like highly processed sugar, not just in donuts, but everything that's become embedded in the food chain since we decided fat was bad, actually is changing the structure and causing us to be more stressed out and affecting the impulse controls of our brain in the same way that alcohol and nicotine do. And we think it's by releasing acetylcholine, which is our endogenous neurotransmitter that's much like, it's like the equivalent of nicotine. So I tell you that because I think it's really important, but sugar, just to tell you a little bit more about its hidden properties, is that sucrose is made up of glucose and fructose. So it's actually the fructose component of the sucrose that's driving this. So what are its hidden properties? Well, we think of it as calories, but we think, oh, well, that's only 100 calories, right? But to our brain and our body, it's more like 1,000 calories. So basically, it binds to the hypothalamus, the fructose binds to the hypothalamus, and then it makes you feel hungry. So when you go and eat your next meal, you basically don't feel full. And the other thing it does is prevents the release of ghrelin and leptin from your belly, which are the two hormones that signal that you're full. So these are very significant hidden properties that people would not be aware of. Thirdly, is that it produces so much energy for our body that our body can't handle it. But because our body is a homeostatic device, it has excellent ways of dealing with it. And one of those ways is through the fat cells, which are adipocytes that line our belly and our thighs. And what it does is it creates, it stores the energy, and then if there's too much energy, those cells divide like cancer cells. And so you can see in this picture here, when you lose weight, that those gas chambers shrink, but they never really go away. And then when we regain weight again, they come back again. So I think that's pretty amazing to know that. So what that means is that when you're working out, and then you reward yourself with a Frappuccino, well, mine was sugar to go to after my marathon running, then basically you've got to work out for another 10 times just to get rid of the reward of the Frappuccino. Make sense? It's really annoying. But anyway, so this happened to me personally. When I was training for marathons, I was actually, you can imagine what happened when I saw all these results coming out of my lab. But I was training for marathons at the time. I was running 19 kilometers. I was really struggling to lose weight because I'd had 2 children, a really stressful lab, and I'm not going to go into all the details of that. But anyway, needless to say, when I got back to Brisbane, I was very unhealthy and very overweight and really struggled. And then when I got these sugar findings, I was like, sugar's my go-to. So I started to reduce sugar, I started to stop sitting as much, etc. And then I started to get my waistline back again. So sugar does do these things. I also started to get my appetite, I started to feel full again after eating meals. And that hadn't happened to me for like 10 years. So the next thing I want to focus on for you is the fact that I've been studying how alcohol and sugar and all of these things change the brain, right? But then I discovered the work of Ender and Folletti, and basically people use alcohol and sugar as medication. So I'd been developing medications for medication, and I decided that I think I may have got a little wrong. So I had to re-gear my lab, and that's because the great understanding that adverse childhood experiences, which we like to call ACE for short, everyone has an ACE score. Lucky you, including me. I can tell you mine because that's how I am. But basically you can learn about it at acesstudy.org, and I'm happy to send those links to you. This work has been done 25 years ago. It's been replicated all around the world. Let me just show you some of the implications of this work and why I've retooled my whole research lab. Basically, what they went on to show in people that are college educated, this is Kaiser Permanente and the CDC that ran this study. They looked at, it's basically a survey where you answer 10 questions, and I'll show you a couple of them. The answer is yes, you get a 1. If the answer is no, you get a 0, which means you end up with a score between 1 and 10. And what they went on to show was the higher the number of adverse childhood experiences, the greater the impact on brain development and health. That then leads to predictions for anxiety, depression, addiction, and all of these things later in life. So these are the kind of questions that they asked in the survey. Pretty, not too bad questions, like did a parent or an adult in the household swear at you, etc. I'm not going to go through it, but let you know that this is completely publicly available. And this is the thing that drove me to look at this graph. So these are college-educated people. These are not people in trauma and violence and poverty. These were done with people, 17,000 people that had healthcare membership in California. And I think it's pretty clear to show you that the greater the number of adverse childhood experiences, the greater your score, the much greater susceptibility of developing alcoholism. I can show you this for depression, and I can show it to you for obesity. I can show you that basically 80% of women that are IV drug using have high ACE scores, and most of it driven by sexual abuse, for example. So we now have neuroscience, brain imaging technology, where we can actually show you the brain for the first time. And that's the difference. Because when you're working out in the gym and you're doing your biceps, you can actually see, I need to do more, but I do. You can actually see your muscles changing, you can feel yourself changing, and so you get rewarded for the work you put in. But if I tell you you can change your brain if you wake up in the morning and think of 3 things you're grateful for and do that for 28 days, you're going to tell me to blow off. Right? Because you just don't see the changes happening inside your brain. And I think that's the difference. I think knowledge and education and brain imaging technology and the digital revolution are coming to disrupt mental health. And we need to do something because it's not getting any better. So I'm here to tell you that each of us have the capacity to be the boss of our brain. We just have to want to do it. And that's the difference. You're going to say, what's the link between adverse childhood experiences and what I'm showing you here? This is a brain, sorry, I meant to point that out. So on the left-hand side, on the right-hand side is a normal brain. We're looking at the red for looking at activity in the brain, and on the left-hand side is someone that's suffered from extreme abuse. And you can see deficits in brain activity in those parts of the brain. What that means is, from a human brain perspective, stress actually takes down through neurochemical mechanisms the communication channels that sit up here in the modern human part of the brain, which is the prefrontal cortex. And those parts of the brain are involved in executive function, which is impulse control and decision making and behavioral control, for example, also empathy and compassion. So you can see and wonder, why does our brain do that to us? Because it's absolutely crazy, because we should have evolved sufficiently to overcome all of this, right? Don't you think? We're meant to be clever. Okay, there's a prize here. Can anyone guess what's going on? Yes? That's right. Clever Adelaide, thank you. Oh, who was that? There's a prize for you. Can someone pass that to her for me, please? Oh, so a snake. So basically the cat thinks it's a snake and jumps, right? Now, don't go home and try this with your cats, because I know it's not going to work because you have loving homes, right? So you have not stressed out cats. So they're going to sit back and go, oh, it's just a cucumber, and walk around it. You know, that's what Adelaide people do, I know. So basically what's actually happening, so the reason I show that video is because it's a beautiful demonstration. We have the same circuit going on in our brain, right? So this circuit is set up for our survival. It's really, really good. It's so fast, it happens in microseconds, milliseconds, such that the cat only has to look at something and think, oh my God, there's a snake, I'm gonna get out of here. Right? So it just flees the scene straight away. That's driven by a circuit in the emotional part of the brain. You've heard of the amygdala, but it's also tied and wired to the nucleus accumbens and all of these amazing places. They're so old. They're so old, and they're set up for our survival. So this is why I'm so passionate about what I do. When you understand that, I don't want to live in my cucumber. But anyway, so this part of the brain is so ancient. It goes right back in history. For that very reason, because we want to flee away from things that make us scared. It's also, the thing about it is, because it's so old and so hardwired, unlike our human brain, which is taken down by stress like that, this part of the brain loves it. So this part of the brain, in the bottom middle part of the brain, is actually prioritizing stress over happiness, because its primary job is to keep us alive, and it's done a great job, because look, you're here tonight. It just doesn't do a great job at driving healthy habits for us, right, unless we have to drive them in ourselves. And that's the principles of neuroplasticity that I like to talk about. So let's just look at how old this thing is, just to get an idea. When you actually get to see and feel that, you're like, wow. And that's why it's so hard for us to evolve out of it. It's doing a great job, and we're in a modern world where we don't have to flee from snakes and, you know, all of these ancient tigers and all of that. Now all you have to worry about is superannuation and house prices and do the kids go to good college and, you know, all of that stuff. So if you look underneath underneath a tree and you look inside the root of a tree and how it uptakes nutrients, and you look at the synaptic vesicles. So we have vesicles inside our brain. That's how our neurons communicate with each other. They've adapted it from the same kind of apparatus. I can go back to mushrooms. Don't go down the psychedelic pathway. I was asked that on ABC radio today because there's tons of psychedelic neuroscience happening in America right now. But if you look underneath a mushroom, these mushrooms can communicate to each other across miles. They can turn a rainforest into a meadow, and people have shown they actually have action potentials when they're doing that. And what does our brain do? How does it signal? But it uses the same apparatus. Now, how old are mushrooms? How old are mushrooms? Like, they just followed bacteria. So that's like forever, right? As far as we know, since stardust. So basically, if you look at a bacteria, okay, you're going to say, we're not bacteria. And I'm going to say, we have some bacteria inside us, because why do antibiotics not work? Well, the bacteria form biofilms. They're so clever. And those biofilms signal across a network using chemicals and peptides that we have in our brain. And now we know the microbiome is like producing serotonin, is affecting our mood. So everything's connected. But we're still just humans. I know we think we're really clever, but we're kind of not in some ways, because we still live with mushroom brain. Anyway, and you're going to say to me, I don't believe you, which is fine, I don't mind. So let's just do a little test, because in Adelaide I know you're all mathematicians. So we're going to do a math test, and it's really straightforward. This is called a working memory test. So what I'm going to do is give you a math problem, and then I'm going to give you a sequence of numbers, and then you're going to say back to me the answer to the math problem, and then you're going to turn to your neighbours and do the sequence of numbers in reverse. So you're not allowed to use your iPhones. You're not allowed to write it down, because we are in Adelaide. Brain power. Okay, ready. So the numbers are 2, 5, 7, 8, 9, 3, 1, 4. 99 plus 3 is? Say it out loud. Great. Now turn to your neighbour and say the number in reverse. Come on, ANS, you can do it. Okay, who would like to have a go? Because there's another prize. Go for it. Help each other. Go. No. Just wait, start again. No. We're in reverse. No. No. 4. Someone said 4. 4. Do you want to go for it? No, not quite. Close. No, not close. You got 4 and 1. Woohoo! I knew Adelaide was going to do it. There you go. Thank you for doing that. Appreciate it. So I'm going to ask you, what do you think was happening inside your brain then? So immediately I go math test, you go, I don't do math. That's a freeze, right? So first of all, I stressed out your brain by saying math test. So sorry about that. That's a negative biohack. Don't blame me for that. And then basically, you either froze and didn't even try and said, I'm not even going to bother. And then the second thing you might have done is like, I'm here to hear about my brain and how I can treat addiction. Why has she been making me work? Why is she making me like have to show people next door that I'm not really smart with the numbers? Or the third thing is, I'm just going to go to the bathroom for a second. So they're the 3 responses, right? Freeze, fly, fight, basically. So you can see how that little amygdala in there is shutting down your prefrontal cortex, right? So what sits in the prefrontal cortex but your working memory, and that's your ability to keep 8 pieces of information in your brain and then repeat it back in a different way, right? So that's what we're doing all the time, every day, is listening to instructions and then trying to follow up on them. But when you're stressed out, that does not happen. So, one of the main big biohacks is like putting your arms up, being calm, and then going, oh, math problem, easy. I'm just going to sit back and think about what she says, and there's no problem for me because I'm really clever. And then your brain's nice and calm, and so your prefrontal cortex and your working memory will keep all the pieces of information in there and allow you to bring it back. So I think you can immediately see that if kids are coming to school from very bad situations and then we're asking them to sit in a classroom and take in information as if they're coming from a Mozart home, playing Mozart every morning to them, that we're going to have different outcomes. So the bottom line is we're not born a blank slate. Sorry to tell you that. We're not playing off a blank slate. So we're coming into this world completely pre-wired. And it's from millions of years of evolution. Yes, we have a genetic blueprint. That's why you do what your mother does sometimes. Yes, we have epigenetics, so your brain and your body grows up in an environment. If you're growing in utero and you have a stressful situation, it's going to affect your brain development. If you grow up in a loving home, you're going to have a much better chance at not getting addiction and depression later in life. But we now have evidence, and this is the beauty of science, why I just love what I do, is we now have evidence that non-coding RNAs and microRNAs are actually, like from the sperm, for example, are affecting the way that genes are expressed that lead to diet-induced obesity for 3 subsequent generations. And so we're going to have the potential in not too distant future, because it's happening for like these rare diseases like SMA, where they're doing genetic engineering using antisense oligonucleotides to actually help kids that have SMA, this spinal muscular atrophy. It's the first time in history that that's happening. I can see that's going to happen in this space also. And I was just talking to some people in this area. So we might be able to have the opportunity to do genetic engineering on microRNAs and non-coding RNAs to actually improve the chances of all this, I'm not going to swear, I'm not allowed to swear, multi-generational stuff that we inherited. So let me show you this panel. So this is different types of mothering. What we're looking at there is gene expression. So same genes, but in red means those genes are turned off. In green, they mean the genes are turned on. Same genes, but look, totally different expression from 2 different types of mothering. And it's not all about the mother, guys. Trust me. It's all about the sperm at the moment this year. It is. Yes, finally. Sorry, women on the rise, women on the rise. Okay, so this is, I know, sorry. Anyway, back to grounding. Okay, so the reason that I came out of my lab is because of neuroplasticity. Neuroplasticity is the ability of our brain to change. So I don't want you to go home depressed and thinking, oh my God, microRNAs, God knows what I got in my history. So the hope and the thing I absolutely know is that the brain can change forever. There are women doing triathlons at 105. There are men changing their bodies at 90 and winning 200-metre sprints at 93. They were not working out at 89. They won a 200-metre sprint at 93. I know of people that have come from extreme trauma and totally changed their whole mindset and life. And that's the total principles of neuroplasticity. The brain has a massive capacity for change. And it's in the things you can't see too. Like we can't even measure some of this stuff because it hasn't even grown into your brain yet. That's the amazing thing about neuroplasticity. Jill Bolt-Taylor's TED Talk, she rewired her brain from stroke. And the thing that upset me the most, but I love it, is Norman Doidge's book about the brain that can change itself that everyone knows, but then they think it's only about extreme things like, like learning difficulties or stroke. No, this is about daily living. I want and hope for each of you to be happy, healthy, and strong, because I think you all deserve it. And This is why I moved into this space. It's what I work on in my lab. We're doing it from every point of view you can think about, from the very basic science all the way through to me giving these lectures and starting podcasts, so that you can take this information right this second and wake up tomorrow morning and start differently, because the brain will do it for you. It's just simple, it's just not really easy. So the question, can we reboot neurogenesis? Can we understand neuroplasticity from a molecular level? And this is some very recent work from my laboratory, where we actually used an anti-anxiety drug called tendusperone from Japan, and we were able to show that we could actually, through a very novel mechanism involving serotonin, we could actually help to prevent the loss of brain cells by promoting this particular receptor called 5-HT1A. So yes, the mechanisms are starting to be worked out. But for you in the audience that came out tonight, which I'm very, very grateful for you today, for your family, for your grandchildren, and for the future, it all starts with your mindset. I took 3 years to believe it. I didn't believe it. She told me about neuroplasticity and I blew her off. And then I had my own things happen, and I was sitting in my lab writing a book chapter on the molecular mechanisms of brain resilience. Your brain can learn anything if you let it. So basically it all starts with wanting to do it. It starts with your mindset, and it starts with you understanding that you have brain power that you're the boss of. Now, I hope that I was able to make it clear that your brain's the boss because of evolution. I hope I made that really clear. So for you to become the boss, you have to drive in the things that the brain's like pushing you against. Brain loves negative stuff. Damn it. It just does, because it's wired there for survival. So for us to change the trajectory to be happy, healthy, and strong, we have to drive those changes with the daily things that we do. That starts with knowing the brain's incredibly plastic, that you have complete responsibility and accountability to take it forward. Yes, there's a lot of stuff that happened. Yes, you can't change it. Yes, all of that. But yes to the future, because I think we only have one precious life, and don't you want to wake up and feel fantastic most of the time? So it all starts with nutrition and exercise and water, but also you're having a really wicked morning routine. Now, why I say that, if you lie in bed tomorrow and think of 3 things you're grateful for, which sounds a lot like Buddhism and mindfulness, but basically it will drive positive neurochemicals inside your brain, and that's been shown scientifically. So all you have to do is try it. And if you can't do it for 3 days, don't expect to change your life and to get fit and healthy, because it's not possible. So we have to start with simple things first, and then repeat, because the only way you get these new synapses and to reroute things inside your brain is it takes those synapses being pounded upon, just like when you're learning a new language, and then eventually those synapses become hardwired. And you know what the cool thing is? Your brain will do it for you then. Once you've formed some of these habits, you're now using the magnificence of the brain machine to drive those habits for you. So next minute you know, after God knows how long, depending on how reactive your brain is from years and years of mushrooms and stuff, it could, I'm serious, so for me it took years. I had a big mushroom brain. But anyway, eventually, with practice, these things will become automatic. So next minute, you know, you're waking up and you're putting on your running shoes, or you're going swimming, or you're really aware of how much sugar you're eating or how much alcohol you're drinking. And like, how do you know? Like, how do you know you go for a Mars bar when your boss really annoys you, right? That's what I used to do. And I had no idea that it was like worth about 900 calories and 4,000 hours at the gym at the time. So I understand this sounds really simple. Why are we all still suffering? Because that's just the way it is. But so we need to help everyone, including myself. And why don't we take the power of technology and make something good out of it? So we've built a game called Trace It, which is still in production. We're developing digital technology to enhance brain, to bring in big data and deep learning and other mechanisms that we can apply to help people break this hugely hardwired stress-reward cycle inside our brain, because each of our brains are completely different and each will derive different things that they need to drive these changes. Also, we're studying at the molecular level, how do we drive these restorative activity? Because basically, this is one of our neurons from my lab. We can label those neurons and we can study them, we map them. I just love them. I've loved them forever. Sorry, ex-husband, no. Just joking. But the interesting thing is that you can't actually see some of these changes. So I can measure and map what I can see under the microscope. But with neuroplasticity and brain training, we're going to be driving new synapses. So we can't actually see them until we have those brains trained. If you see what I mean? You don't know what you don't know. So I believe there's a middle way coming where we're going to combine the best of modern neurosciences with the best of ancient practices combined with genetic engineering, where we're going to be able to tap into some of these microRNAs and make changes that are going to help prevent some of the pre-wiring that's come in to make us really unhealthy. I really believe there's a middle way, and I like to think of personalised medicine as a tapestry that draws all of these things together to make your lives more happy, healthy and strong. I also started a podcast because I think this information can get to the public and make a small difference. Even if I just plant seeds, that's really good enough for me. And it's really made a huge difference in my own life. And so I just want to be able to share that information because we've got millions of papers already on different parts of this subject. I wrote a book called Smashing Mindset. I'm in the middle of writing another book from all my learning since this time, because we keep learning and changing. Science is just magnificent. It's bringing in amazing big changes, especially with metabolomics and microbiomics and epigenetics and non-coding RNA. All of that information is coming to us and we're really making big advances. So I'm really excited about that. So thank you for coming. This is built on the backs of tons of people that have worked with me over the years, both here in Australia, in America, at UCSF, and just a whole lot of other people that aren't even on this slide. And I just want to thank you for coming out tonight and listening to a brain talk on a Tuesday evening. So thank you.